So this is AP Bio Day, what day are we on? 12? AP Bio Day 12, we're on Thursday, January the 21st. So tomorrow, which is day 13, especially for you guys at home, we will be conducting a lab in class. Now, it'll either be me or the lady from Science in Motion. I've actually never done this lab before. I've done similar labs. This one's set up a little bit differently. But we will be doing this in class. I will record it for you guys at home so you get an idea of what's going on. And she's going to do it for my first period, and then she may just leave it with me for you guys. It's really not super complicated. And I'm going to go through today and kind of give you the high points of what to expect tomorrow and the background, knowledge background, so that you have some idea of what's happening and why it's happening. So that's what we're going to be discussing today. First thing I want you to look at is the lab packet that I gave you guys yesterday. So this was on, I think I posted this for you people at home on Schoology for either day 12 or day 13 to read over. Didn't I tell you guys to read this yesterday? Yeah. Okay. What you're going to do is, I've got at the bottom, annotate the lab write-up and write 4 big takeaways from it. We're just going to add that, wind up stapling that into your lab packet that you just got. And then that will be all turned in together, and that'll make my life easier so that everything's in the same spot. Okay? So what are some takeaways you got from reading this? What is the lab about?
It's about HNPCC.
What is that?
It's like a type of inherited cancer.
Okay, so it's on colon cancer, right? And your colon's also known as your, what do we call the colon? In your belly. Y'all got to get out more. Large intestine. So you may have heard of like a colonoscopy where they stick a probe up there to check things out and whatnot. Usually what they're doing is looking for polyps. They're looking for anything that's cancerous or precancerous. And some of you out there on internet land may know someone who has colon cancer, or if there's a high likelihood of having cancer. One thing that you should have taken away from this article is that there are some genes that can make it more likely for you to get colon cancer. And so that's what we're looking for in the lab. Seeing as most of you don't have a good idea of what's going on, make sure you read over this before you get here tomorrow, or otherwise you're going to be very confused and behind. So make sure you read through these two. Do the assignment at the bottom. Just annotate as you go through the important parts. Make sure, I want you to give me 4 big takeaways from this. And then this is what the lab's on tomorrow. So we're looking for some specific gene traits in the lab, okay? So don't put that away. We're going to come back to it in a second when we talk a little bit about how the lab is going to be conducted. At the end of class today, I'm going to have a segment on plasmid mapping. Plasmid mapping is one of the more complicated activities that we will do in here. They have had these on the FRQs before, and so it's something we need to practice. For those of you who are in my class online, I think I posted an activity on day 10 or 11, a plasmid mapping activity. For those of you who are in class, you may want to go back and check that out. There's some more, it's just basically a bunch of practice examples. And go back and do some of those if what we do today is not enough to whet your thirst for plasmid mapping. So it's going to be complicated, and I'm sure you guys are going to figure it out because I'm going to lead you through the process and teach you how to do it. All right, so let's finish up Biotech Part 3 today, and then we're going to work our way through Biotech 4. So Biotech Part 3, we are talking about bacterial cloning mechanisms. Bacteria, why would they want to clone themselves? What do we call that?
Binary fission.
Binary fission. Why do they clone themselves?
So that they can reproduce.
Okay, so that's one of their methods of reproduction. And when bacteria, can bacteria have sex?
Yes.
Sort of. They can exchange what with each other?
Plasmid.
Plasmids. So what we're loosely calling bacterial sex is basically the exchange of plasmids, little small circular pieces of DNA, between a structure called the pilus or pili, and in doing so, they become a little bit different. Sometimes they get like antibiotic resistance or other things that are helpful to them from one of their partners that they're exchanging DNA with. So, once they exchange that DNA, then they're going to start reproducing through binary fission again. So really, they're exchanging DNA, and then they clone themselves out afterward. And if that gene is something that's effective and good, then they will keep passing it on. If it's not, they'll die out. And so it kind of leads into when we get to evolution is really all going to be about genetics and the environment. So we will come back and talk about a lot of this stuff. Let's talk about on page 413. Yesterday, or a couple days ago when I was absent, I think you guys are supposed to read through this, how bacteria clone themselves. On 413, let's talk about how bacteria fight off viruses. What do they use?
Oh, they use restriction enzymes.
Okay, so they use restriction enzymes to do what to the viruses?
Slice them up.
Yeah, slice them up. So, how do they know what to slice up? Well, they're looking for specific sites. So certain viruses infect them, they've cut them up, they figure out certain sequences, and then they're able to remember that and then cut them up at the same spot next time. Now, do they cut up the DNA evenly?
No.
No. You've got to leave a what?
Sticky end.
A sticky end on the DNA in order for it to do what?
Grab on.
Yeah, something to grab onto. Now, since the bacteria is going to save some of this viral DNA so it can fight it off next time, it needs to save and embed part of it in its DNA. And so here's a picture. You can see right here the sticky ends where it sticks out. So it's not cutting that DNA evenly. It's leaving a little segment for it to for the DNA to be cut and pasted in to some other DNA. So you don't want it hooking together like this. You want it overlapping so that they can stick together and that they can all be hooked together very easily. Okay. Number 1, Matt, read that for us. On 413.
Restriction enzyme cuts the sugar phosphate backbones at each arrow.
Okay, number 2, Mariah.
DNA fragment from another source is added. Base pairing of sticky ends produces various combinations.
Now, we have hijacked this mechanism, okay? So we've hijacked it, and we figured out these restriction enzymes, what they can do, where they cut stuff, where they cut the DNA. And so we talked about it in terms of like production of insulin or human growth hormone or something of that nature, where we want to cut a gene out of a human and we want to paste that gene in a bacteria. So in order to cut it out and in order to cut that plasmid, what do we have to use? Restriction enzymes. Which enzyme, what do we know about the restriction enzymes you cut it out with and the ones that you cut the bacteria with?
You have to use the same one.
Why?
So that they make the same sticky ends. So
Good. You got to use the same restriction enzyme so they leave the same sticky end so that they can attach. Very good. So, and you're going to see some restriction enzymes that are used in our plasmid mapping activity today. Number 3, once they get cut and pasted in, then what is used to tie all that back together, stitch those DNA back together? Ligase, the same enzyme we used in, or that was used in what?
DNA transcription.
No, transcription, good. So when, not even transcription, DNA replication of the Okazaki fragments. Remember, they had to be stitched back together. So ligase used same enzyme used there to stitch that DNA together. All right, turn over to page 414. So hopefully on part 3, that kind of clarifies most things. They talk about genomic libraries where you have these libraries of stored plasmids and genetic information that have already been made, and they basically just use bacteria to make a bunch of copies of those once they do that so that they have those for future use. On the bottom part of the notes there on number 3 of number 3, it talks about using reverse transcriptase and making complementary DNA. This is that process where I told you, you cannot take a piece of DNA and cut it out of a eukaryote and put it in a prokaryote. Why?
Because prokaryotes don't have introns.
That's right. So they're not going to know what to do with those introns. So you have to first take that DNA from a eukaryote, convert it into mRNA, get rid of the introns, and then use reverse transcriptase to convert it back into DNA, free of introns, and then you can cut it and paste it into a bacterium to use. And so that's what part 3 is about. And part 4 talks about yeast artificial chromosomes. A similar process there for recombining in yeast. Yeast are kind of a simple eukaryote that we all use for bread production and some people for making alcohol. But you don't really need to know a lot about that. All right, part 4. All right, the lab tomorrow. So how many of y'all have ever seen Maury? You know, like, Who's My Baby's Daddy? You know what I'm talking about? Ever seen those episodes? You're too good for that? Mariah is the only one that's like me and watches that crap. All right. So it is depravity at its finest. So in what you're going to have tomorrow is on basically like the one that's going to, or similar to the one like on 414. So there's different gel setups that you can use, but they're all have the same components. Let me show you here. So at the top of the page here, we have these gel chambers, and then in the middle of the page, you can see what it looks like after the DNA runs through there. So at the top of the page, basically what you're going to have is you're going to have some kind of little chamber, and you're going to have a gel that's like a thick jello. And in one end of it, you're going to have these wells. What's going to go in the wells? So basically, you've dug a little well out into the gel, but it doesn't go all the way through. So what's going to go in those wells? DNA. So in some cases, if you're on Maury, you might be doing this to find out who's the baby's daddy, like which DNA matches up with the kids and the mom, and which one, has the same DNA going with the dad, to find out who the dad is. In the cases of the lab tomorrow, that's why y'all need to read through that. It actually discusses the determining if they have that gene or genes that lead to may lead to you developing colon cancer. So that's what we're going to be looking for tomorrow. Okay. So you take the DNA, you insert it in the wells. Now, which way does electricity move? Thank you. Which way, Katie? Negative to positive. Good. So electricity moves from negative to positive. DNA has what charge? Negative. So you can see in this well chamber right here, it's negative on the left side and the wells are on the left side. What is that negative electricity running through there going to do to that negative DNA? Negative and negatives make a... No. What? Repel each other. Thank you. It's like, think magnets, but electricity. So it's going to repel each other, and the DNA is going to do what?
Go towards the end.
It's going to go toward the right. It's going to move down the gel toward the positive end. Good. So, DNA. Are you just going to throw your 46 chromosomes in those wells? No. That'd be too big, and they'd pretty much be the same in everyone. So what you're going to do is you're going to take what they do is they take DNA and they chop it up. So they chop it up using what, Ashlyn? Restriction enzymes. So those restriction enzymes are going to cut the DNA at specific known sequences. Okay. Now, everybody's DNA varies a little bit, unless you're identical twins, and then maybe there's some methylation, but the DNA is pretty much the same. Okay. So DNA is going to travel at different rates, and it's going to spread out depending on the fragment size. Okay, remember this is like a thick jello. Okay, this gel that it's moving through, but it is porous, so stuff can move through it. So the smaller pieces of DNA are going to do what?
Move through the pores.
What?
Move through the pores.
The smaller DNA is going to move slower. Why would it move faster? Smaller, it can move through the holes, right? I mean, there's small little pores in the gel, so smaller the DNA is, the faster it's going to move through the gel. The larger pieces are going to go slower to get through there, okay? Now, can you tell how big the DNA is that's moving through there? Yes, you can. How could you tell though? Like, how could you tell like how big, like how many base pairs the DNA is? Like how many A, T, C's, and G's? Because the longer the electricity is on, what's going to happen? What? Yeah, the further it's going to move, right? So the longer you leave the electricity on, the further the DNA is going to move down the gel chamber. Okay. So how could you tell how big the DNA is? You can't put little numbers on the side of the gel chamber, right, and say if it goes this far, it's this big, and if it goes this far, it's this big.
Make the chambers increasingly smaller. So
What?
if you make the chambers increasingly smaller, the farther it went means the smaller it is. Because
Well, no, I mean, we know that. We know the farther it goes, the smaller it is. But we need something for comparison. So if you look here on 414 in this bottom picture on B, you'll notice it says standard sizes. So basically what somebody's done here is they've taken some DNA and that are of known base pair sizes. So like one is maybe the one on the right's 5,000 base pairs and the one on the far left is maybe 40,000 base pairs. And so you know this segment is this big, it travels this far. You can go over to these other segments that you don't know and you can, like, well, is it next to it? Well, it's the same size because it's traveled the same distance, okay? So that's what the standard is there for. It's a method of comparison to see how big the DNA is, okay, that runs through the gel chamber. Now, in your notes, let's skip on down to where it says gel electrophoresis. It's called this because it's going through a gel and it's using electricity. Part A and B, read that for us, Katie.
This process is used to create a DNA fingerprint. Take different DNA samples and expose them to the same restriction enzyme to cut the DNA into fragments. This creates restriction fragment length polymorphisms, RFLPs. These create fragments of DNA having different lengths.
Okay, all that said is you're creating a specific DNA fingerprint by looking at your DNA and how it spreads out in that gel, okay? So it's going to create these little fragments, and it's going to basically be like this DNA pattern is going to be unique to you based on where it's cut and what your DNA looks like, okay? And you can compare that and see, like, if you were wanting to know if you were somebody's baby's daddy, then is like half your DNA matching up with your child? You know, as if you're the mom, how much is half your DNA matching up with the kid? So that's what you would be looking at. How many of those fragments are similar? Okay. Is there a certain length fragment that identifies for a certain gene, like a gene maybe that causes colon cancer? So, good. So basically how this works is you turn on the electricity, it moves toward the positive end, and then smaller segments move quickly. If you ever had to do this in college, basically what you would do is when you're done letting it roll through there, you would have to stain it. You put this piece of like paper that's got, or there's a liquid you can soak it in, and it turns the DNA like a bright purplish blue color. And so you can see it. I don't think, we're not going to do that with the ones we have. I think it's maybe going to fluoresce, if I'm not mistaken. I will post this after we do it tomorrow. I actually have not conducted the lab the way we're doing it tomorrow, so it will be new for all of us, although it's pretty much how I'm telling you it's going to be, okay? We talked about the Human Genome Project earlier. Basically, this is where, like, scientists said, hey, we're going to map out all the A, T, C's, and G's in the human genome. And it took years. Why did it take years? There's billions of them. And the process to actually figure this out using this DNA microassay at the bottom here is not easy. I'm not even going to get into it with you guys because it's just confusing. I don't think you really need to know it. But basically you're assigning each molecule, different molecule, a different color, and then you go through and it maps it out, and a computer has to map out like where everyone is. When you're talking about 6 billion nucleotides, I mean, that's a lot of A, T, C's, and G's to go through and write down on a paper somewhere, right? So anyway, basically what they did was, like, we got Walker over here in Switzerland. You're going to do chromosome 5. Matt, you're in Germany. You do chromosome 3, and so forth. And so basically they divided up the DNA, and they said, you do this part, you do this part. And then the scientists came together as part of that project to figure out what that genome looked like. Okay? On the back side of part 2 here, it talks about uses for gel electrophoresis. in pharmaceuticals to help develop vaccines. It could be used in gene therapy. We talked about that in terms of bacteria, modifying bacteria for oil cleanup. DNA technology used for agriculture, like they make bigger food, pest-resistant food, meatier livestock. So all these things are applications for DNA technology. You guys, we even talked about in the cloning video. Y'all watched that, right?
Yes.
Where, you remember the pigs? What were they trying to do with the pigs?
Trying to make them so that the organs were able to be harvested and like be able to put in humans.
Yeah, so pigs basically have the same size like body organs that we do. And theoretically, you should be able to take a pig heart out or whatever organ and put it in a human, and it should last like 10, 15, 20 years if you take care of it. So the problem is you can't do that. Why? The sugar's on the outside of the heart. And that sugar says, hey, I'm a pig, and if you put me into a human, then it's going to kill it. Okay? So at some point, I hope we get to the point where we can use this DNA technology like CRISPR, where you can cut that sugar off, you can replace it with a human gene, and then you could use that for organ transplant. So you can basically get rid of all these organ transplant lists. You don't have to wait for people. Somebody did make a good point, though. Well, if you do that in a pig, you would have to modify a lot of their external sugars, especially in their immune system, because you wouldn't want them to have a heart that said, I'm a human heart, and then be in a pig, and then the pig kill its own heart. So you have to you can't just modify just one thing. You have to modify multiple things in the organisms.
Are all the pig organs like similar to ours?
I think they're pretty similar, yeah. Enough where they could be used, if not for that. Physiologically, I think so. And they chose pigs, I think, because that's something that's similar size-wise, not too far off. And then, they're easy to grow. They're relatively inexpensive. And so it would be perfect if we could, do that. Well, not perfect, but pretty close. All right, the next thing, transgenic organisms. 2 different organisms combined into one. You might also call this a chimera. Man, there's some weird science tech stuff going on, sci-fi kind of stuff going on with this. I saw an experiment a group did somewhere overseas. I can't remember where it was. You'll have to look it up. But these people mixed in, is either like human and mice DNA or human and pig DNA or pig and mice DNA. I can't remember. It was like 2 different organisms. I want to say they put some human cells in there. Could be wrong. You'll have to look it up. But anyway, they mixed them together very early on at the embryonic stage when they're still stem cells. And then they started developing, and like both of the cell lines, like if it was human and mice, like they both started developing. It wasn't like one was trying to kill the other or whatever. And so all I could think of was like, if they kept this going, would this be like some weird mouse human person? Or, you get the Batman, actual Batman, or what happened? So like, that's a worry. Like, what if some weirdo scientist in Siberia is like, hey, let's see what we can come up with here. Let's see if we can make an actual Spider-Man. That is a worry. So yeah, have to keep an eye out for that. And genetically modified foods. Foods are constantly being altered to be more productive, more pest resistant, and use less resources. So there is benefit in that. Let's back up here real quick on 415. Okay, so on 415, in your notes at the top of part 4, where it says polymerase chain reaction. So, PCR, you need to know this in the book. Now, let's talk about COVID. Oh, I need a drink. I'm going to be honest, I'm tired. I don't know what's up with my dog, but she got up like 6 times last night with diarrhea, like once every hour. I had to go outside. I saw her outside squatting down, walking around the yard, just hoping something would fall out. And then she'd come back in, and then like an hour later, she'd wake me up, and my wife never woke up. It's like worse than having a baby. And usually babies don't get up that much. I think she fed her like some kind of steak bone or something. Must have been disagreeable. Stop feeding the dog weird stuff. And I was just sitting there like leaning against the door, waiting for her to come back in, to open it back up. And I was like, And now y'all are catching the worst of me. So, COVID. I don't know. I probably shouldn't ask you if you've had a COVID test, but let's say that you've had a COVID test. How does that test for COVID?
They stick up, like,
No, I mean, they stick the thing in your nose and they're getting some DNA out, right?
Right.
All right. Well, do you think there's a lot of DNA in there or a little bit?
a little bit.
Probably just a little bit that they're able to actually get out. So how do you make more DNA from a small amount without using an organism?
Go through DNA replication.
Good. PCR, polymerase chain reaction. Who was it figured this out? Carrie Mullis. I don't know if that's a guy or a woman. I guess it matters. 1983 here. And this person figured out that if you, let's just read through it here. Number one, Miranda.
Polymerase chain reaction.
No, in the book.
Oh, okay.
Sorry.
Denaturation heat briefly to separate DNA strands.
Okay. So all they did was heat the DNA, and it did what?
Separated.
Separated. Tay, read the next one.
Annealing. Cool to allow fragments to form hydrogen bonds with strength of target sequence.
All right, so they heated it. They cooled it. Number three, Mason.
Extension. DNA polymerase adds nucleotides to the 3 end of each primer.
Does that sound familiar, DNA polymerase?
Sounds like RNA polymerase.
Well, DNA polymerase we talked about in replication, right? It just has nucleotides. So basically all you do is you heat the DNA, you cool the DNA, you make sure that you have plenty of nucleotides mixed in there and also some RNA polymerase or DNA polymerase enzyme mixed in there. And in the end, you wind up with,
4 sets of DNA. Or 2 strands.
well, I mean, you started out with 1 and now you wind up with 2. And then the next time, what happens? Yeah, so what type of growth are we going to see here? So you have exponential growth every time it's doubling. So, leads me back to COVID. So basically what they do is they'll go through and cycle this DNA and make copies. And I think the normal is about 30 to 35, somewhere in there. Maybe it's closer to 30. And imagine like cycling this and doubling it 30 times. I can't think of how many millions of or more strands you would have, but it would be a lot, even if you just started out with very few. Now, there's a problem with this. If you cycle this DNA enough through PCR, sometimes if you cycle it and basically make a bunch of copies enough, then it'll almost show up every time. So what wound up happening early on, and there was a New York Times article, you may have to use the Google to find this, back in the summer that was saying that a lot of the false positives were attributed because they were running PCR instead of about 30 times, they were running like 35 to 40 times. And so basically they were copying this DNA so much that almost like it would show up regardless if there was anything there or not. And so it's kind of like a rigging of the system. I don't know if they were doing it on purpose. They were trying to make sure they got enough, but it was affecting the rate, the accuracy of those tests. So now somebody brought up a good question this morning and I forgot to look it up. If somebody wants to do it for extra credit. What happens after that point? So once they've made a bunch of copies, I don't know exactly how they're actually looking for this, if they've got like a DNA scanner that goes along with this and is looking for a certain sequence. Some of these tests, these rapid tests, they can get them in like 15 minutes or so. So I didn't really look up that part, but this is how they copy the DNA to get enough of a workable sample. The same would be true if, Sarah over here committed a murder and, she looks all sweet and innocent, but like, let's say she stabbed somebody in the back and she got scratched and left one little small drop of blood and there's very little DNA there. And so they're like, hey, let's take a sample, run it through PCR, and we can make about as much as we need. And then we can test that and test it against her blood. So we're really to the point where you need very little DNA to actually do analysis and comparing if somebody commits a crime or something of that nature. Okay? All right, any question about that? So basically, tomorrow, you're going to be doing this gel electrophoresis. You're going to have the gel chamber. She's going to set it up, tell you about it. You guys need to know the background, so make sure you read through that packet. It's posted for you guys that are on my Schoology account. And then I will video as we run through this gel electrophoresis lab and kind of let you see the ins and outs of how this would work. I usually do this with ninth graders when I used to have them, but I mean, it's pretty simple and straightforward. You guys in college one day may do this again if you take a genetics class. So, but this is how you test and compare different DNA fragments. Okay? Any questions? Beautiful.